sure the audio levels sound good, or do they just start out trustworthy?
I can see.
Okay.
On here, so.
Nice.
Can you go move around up there? It's gonna take me like 30 seconds to catch up here. Ashley says go Ricky.
Oh, there's a chat, right?
There
Or
is.
live stream.
Somewhere I should be able to see how many people are in, but I can't at the moment. I'm not going to mess with that actually at the moment.
Just so long as you can see that when it comes time for questions.
Yeah, I'll be able to see. I can see the chat. I just can't see right now how many people are in the. Yeah.
Inconsequential.
Yeah. Okay, and we're at 0 drop frames so far, so actually we're doing pretty well. Okay.
Cool.
So stream health is good. Okay. Well then, hey, we can get this pretty well. Hello, good night. Thank you all for coming out to Ricky Wright's thesis defense. So just a few words before he takes it away. I first met Ricky as he was a cell biology student in my class here at Walla Walla. And that's kind of an unusual time for me to meet students because typically I meet them a bit earlier, but he came to us from Walla Walla Community College, which was I really appreciate that path. He brought an interesting perspective. And one of the very first things I noticed about him in my class is that he constantly asked questions, and really good and insightful questions. And sometimes questions that put me on the spot that I didn't know the answer to, which can sometimes be an uncomfortable situation for a professor. And he kept doing it over and over again. This was a theme that went on throughout my entire time knowing him. I was really happy when he decided to stick around for a few years and pursue a master's work here as well. And he picked up on a project that I've been really interested in looking at RNA editing in octopuses. So I was really thrilled that he was able and wanted to do that. And so he kept up that time. I've noticed one thing about Ricky all the way through is that he's never very afraid to speak his mind and speak his mind loudly and vocally. And I really appreciate that about him. He is someone that lets know exactly how he thinks and feels on things, and that has been both really great and uncomfortable at times, but I've really appreciated it, basically all the time. So anyways, I am really happy that he is here at this point. I'm really proud of all the work that he's done. So with that, I will give it over to Ricky. Maybe clip on you and in the back.
Okay.
On like
Might just pocket it here.
you can pocket there too.
There we go. Cool. Hello. A while back, during my undergrad, I had a conversation with Kurt, and this was probably the first time I'd heard about this cool thing that octopuses do with their RNA, and this was an impromptu conversation. He's just kind of like that. He'll have those conversations with students, and up until I came here, octopuses to me are cool, seen them in nature documentaries, heard about how smart they are, but I didn't feel like they really had much to do with me. And I had that conversation with Kurt, and he was talking about RNA editing. But at the time, I didn't understand even half the words he was using, much less how it all fit together. And I would try and ask questions like I do, and I was completely lost. I don't really know if he knew how lost I was, but boy, I felt really lost. I forgot every single thing he told me in that conversation afterwards. And I was starting to feel like I had maybe bitten off more than I could chew in biology at that point, just that kind of thing. Gave me some serious like imposter syndrome, feeling like maybe this isn't for me. But anyway, my master's research is in RNA editing in octopus rubescens in response to ocean acidification. So clearly some things have changed a little bit, thankfully. The central dogma of biology that we're taught about, especially in like cell biology classes here, states that DNA is transferred to RNA, which is translated into proteins. And pretty much the first thing that they'll tell us here about the central dogma is that it's a drastic of the process. The RNA, in this case, is said to be just a messenger from the DNA to the ribosome to be translated into proteins. In reality, it's not the cases. After it is transcribed, basically breaking the central dogma. But that's fine. When I say edit RNA, what I'm mostly talking about in this case is an enzyme, an adenosine deaminase that acts on RNA. Imagine you have an RNA sequence, like this in blue up here, where it reads ACU so-and-so GACG. That A is an adenosine. Adenosine looks like this, and it has an amino group right here, which ADAR will replace with an oxygen and eventually leave us with a double bonded O. This leaves us with, instead of adenosine, inosine. So you have an I instead of an A in the RNA. Now, when this RNA makes its way to the ribosome is going to read this I as a G. So you're essentially ending up with an A to G edit, even though I is an intermediary. For intents and purposes, it is G. Obviously, that doesn't happen to all adenosines. That would be pretty crazy. So there must be some kind of specificity, an underlying mechanism that dictates that some A's get edited and others don't. That does happen to be the case. ADARs only act double-stranded. An mRNA, they're transcribed as a single strand, but they will fold back up onto themselves and anneal at points of complementarity. And of course, it's not perfect. We see lots of loops here, and any A's in these loops won't get edited by ADAR. And it's possible that those next to loops as well won't get edited by ADAR. Now, this has been tested in the lab. Set and now, just this baseline, this molecule will be edited at a few A's up at this corner. If you compare that to another artificial molecule, L0 down here, it has a lot of extra ribonucleotides added on. And when this is incubated with ADAR, half of the A's will be turned into I's. It's called promiscuous. However, if you change the artificial molecule to have not just a little loop, but a larger loop made by nucleotides that lack to the original editing pattern of the small, short molecule. There's by which you could specify, nature could specify, what adenosines get edited versus what don't. If loops are positioned so closely together, then this could be an underlying layer of complexity in the DNA that is only once the DNA is transferred into RNA. There are examples of RNA editing that are actually adaptive, not just abstract. In this case, there were around the globe at these 8 sites, including Baja California. Here in the coast of California, there was an octopus rubescens, Antarctic octopus, and Arctic octopuses. These octopuses all live in different temperatures of water. Now, they share a protein called a potassium channel in their nerves. And this protein is, of course, a gene found in their genome. When this gene is transcribed into RNA, it won't happen just once, it'll happen many times. So for a gene, there may be dozens, hundreds more transcripts of that gene floating around in the cells. And what they've done is they've looked at the percentage of those transcripts in each of these octopuses that is edited at amino acid position 321. So it changes an isoleucine to a valine by the action of ADAR on this single nucleotide in the RNA. And as we look at the water temperature of the collection site versus the editing levels, what we see is that as water temperature goes up, editing at this site goes down. So remember that, because that's I'm going to come back a couple times. As temperature goes up, editing goes down. This editing, particularly the cold water, because when it changes this potassium channel, it actually makes it more efficient at working in cold water. Potassium channels are important for repolarization of neurons for the firing of action potentials. So when a nerve fires, it has to repolarize the membrane, and it needs to do that quickly. This enables cold water to have their neurons firing quickly. And so it's a cold water adaptation. That is just found in the octopuses that are living in the water. But there are also edits that are responsive in octopuses to changing environment. In particular, there was research done in the past on temperature-dependent RNA edits or temperature-responsive RNA edits. Some octopus bimaculoides specimens were split into 2 groups, control or cold water treatment and water, 22 degrees Celsius treatment, and kept there for a couple weeks. Then, at the end of the experiment, their nervous tissue was harvested, and they took a look at the RNA and DNA and found some edits. So that's when the RNA and DNA doesn't match up. You've got editing. They found a lot of edits, thousands and thousands of them, and most of them were induced by being in the cold water. A couple of interesting ones that they found and looked a little more deeply into, there was a kinesin edit. And if you've taken cell biology, you might have heard of kinesin. It's a molecular motor, basically. It walks along microtubules and carries a load around in the cell. This edit slowed the progression of kinesin along microtubules. That is very impactful and might be adaptive for the octopus, but it's difficult to say exactly how, because there's a lot of interacting factors here. But in a similar vein, there was another protein called synaptotagmin, which is also important in, this is important in signaling in response to calcium in presynapses, which I don't need to go into right now, but in the neurons. And this edit actually changes this protein's calcium binding affinity, which again is meaningful, but we don't need to go into exactly all the meaning of it. It's important to understand, though, that it is a very meaningful edit in response to temperature. Now, I'm sure you've all heard of climate change. The ocean is getting warmer, but not just warmer. As humanity produces a lot of carbon dioxide, that 30% of that carbon dioxide actually is dissolved into seawater. And as carbon dioxide dissolves into seawater, it causes the pH of the seawater, or the acidity, to rise, the pH to drop. Because if you remember what the pH scale is, you take the pH scale and you drop from 8.2 to 8.1, that's an increase in acidity. It was holding steady for a while, and then around the time of the Industrial Revolution, it started to drop a little bit. And at the point we are now, we've got some projections for where we may end up in the future. If humanity really gets its act together, carbon sequestration, stops burning so much fossil fuels and emitting so much carbon dioxide, we might end up on the good gray path, wherein pH doesn't drop too much by the year 2100. We are not on that path, though. We are, in fact, on track for the worst case scenario, the red, where pH is likely to drop precipitously by the year 2100. We see this is the same kind of graph over here, but instead of pH, it's proton concentration, which is maybe a more straightforward measure of acidity that you can wrap your head around. This just shows the same thing, but as proton concentration goes up, acidity goes up, and that is what the graph is projecting for the worst case scenario that we are barreling towards. I'm interested in how does this interact, ocean acidification, with animals in the ocean, in particular octopuses and RNA editing. So I did an experiment. I took 6. I took 6 octopuses and had 3 of them in, no, I did take 12 octopuses. I was going to do a funny thing later. 6 octopuses, sorry. 6 octopuses in control, so regular acidity, regular carbon dioxide, we say PCO2, and 6 in a ocean acidification treatment or high partial pressure of CO2. CO2 determines the acidity of the water. And I kept them there for a couple weeks so that they could show signs of RNA editing or not. And then after a couple weeks, harvested nervous tissue from these octopuses. And then I isolated RNA and DNA from those tissues and sequenced that and used them in a bioinformatics pipeline. That's what I did over the summer of 2022. Here you can see an the experimental setup. So the octopus tanks are refitted ice chests. They are watertight. They have cold water and seawater flow through, bubblers, probes, little dens for the octopuses. And that is where they resided for the 2 weeks of the experiment, whereupon they were euthanized, and harvest day came around. Everyone pitched in, and it was very helpful because I was very worried about that day, very nervous. And I got the data that I needed. I am building this project off of work that was done prior. In particular, my predecessor, a former graduate student of Kurt Onthank, J.D. Sariwit, built a bioinformatics pipeline that I am using. And it takes RNA and DNA data, and I reassemble the RNA transcripts of all of the octopuses into a transcriptome. And then I align the RNA and DNA to that transcriptome and check for mismatches between the RNA and the DNA. And when it mismatches, unless something weird has happened, those are instances of editing of the RNA. And then I compare how much of the octopuses in the control treatment editing versus the octopuses in the ocean acidification treatment. I think it bears mentioning a little bit, when I said Illumina sequencing, what am I doing? I go from RNA and DNA frozen in tubes to digital copies of that RNA and DNA. Now, how do I do that? An analogy can be useful here. Say you have a set of encyclopedias, and you want a digital copy of those. But you're limited, arbitrarily in this example. You have a machine that can read them for you into a computer, but it can only read 300 letters at a time. What do? The only thing you can do, you shred the encyclopedias and feed them to the machine, snippet by snippet. Now, some of these snippets may go missing. Some of them may get misread. So in the end, what you need to do is shred multiple sets of encyclopedias and feed those all to the machine. And the more sets of encyclopedias you feed, the more accurate and more complete your final digital encyclopedia collection will be. And that's what an Illumina sequencer accomplishes. It takes fragments of DNA. The RNA also gets turned into DNA before it gets sequenced. And then using what they call a flow cell, it copies these fragments. And that's how it reads them, by copying these small fragments. And each time it copies them, then it has copies, which it can copy again and read again. So it gets exponentially more data every time it copies these fragments. So you end up with a lot of data, a lot of overlapping data that you can use. And we use that to reassemble the transcripts of RNA from all the octopuses. The original transcriptome was shredded into and read in as reads, and then using a computer algorithm, find overlap between these fragments and join those into contiguous segments, what we call contigs, and then join those contigs, make bigger contigs, and keep going until you've reassembled the transcripts of all of them. Once I've reassembled the transcriptome of the octopuses, then I can essentially locate where everything is. So I take the transcriptome and I align all of the RNA that I got back to the transcriptome and the DNA. And once it's all lined up, you can compare each base and see what is this one, what is this one, what is this one, and then this one reads, for example, mostly A, except for at a few sites, the RNA is reading G. And where the gDNA all agrees this should be A, this is probably a site of RNA editing. I mentioned I'm building on prior work. JD did a similar experiment with this. He used RNA data on his pipeline that I'm now using. And that RNA and DNA data came from a prior experiment that was actually being performed for something else. So this was done on gill tissue. Now, most cephalopod editing so far is dealing with nervous tissue instead of gill tissue. And I wanted to do this experiment on nervous tissue for real this time to see how much editing would we really find in octopus rubescens. Now, compare the prior results on gill tissue versus the nervous tissue that I worked with. Showed 122,592 total editing sites versus almost 280,000 editing sites in nervous tissue. So yep, there's a lot more. Not exactly surprising, but definitely pleasing to see. And of the original, in the gill tissue, of those 122-some thousand edits, only 744 of them were actually edited differently in response to ocean acidification or PCO2 being high. Whereas of my 280,000, almost 12,000 of them were responsive to ocean acidification. And that's a very big number. What do we do with that? Well, we could look at every single one of them one by one, but in the interests of time and efficiency, we decided to filter for the most interesting edits out of those 12,000. And what we did was, in that pool of edits, we decided let's only look at ones that are non-synonymous, meaning since there's multiple codons that can code for the same protein, not all edits do something. Only certain edits will actually change the final protein. We're going to look at those only, and we're only going to look at A to G of those because we're interested in edits due to ADAR. And we want to make sure there's a large difference between the octopuses in control and the octopuses in the ocean acidification treatment in terms of editing, because that's what we're about. And so we set that to be at least 20% difference in the averages of the editing levels. And we used a stronger p-value for our statistical test for this. Instead of 0.05, we're using 0.005. And we also wanted to make sure that we had good coverage for every single octopus that we were looking at. So we made sure there was at least 10 mRNA and 10 gDNA reads for each octopus that we were looking at. What we found was kind of interesting. Out of those 12,000, we found 51 what we call high priority editing sites. And I should talk about the octopus bimaculoides coming in here. So we found a lot of zinc fingers. What does that mean? When you look at well, we don't have a genome for octopus rubescens. We don't know how many zinc finger genes are in octopus rubescens, but a close cousin, octopus bimaculoides, has a genome assembly available, which is very helpful. And if we assume that it's similar in certain ways to octopus rubescens, we can glean some facts from that. In octopus bimaculoides, it has 33,638 genes, meaning if you were to pick a gene at random from octopus bimaculoides, there's a 5.3% chance that it would be a zinc finger. Now, if you were to pick a gene at random from our pool of 12,000 significantly differentially edited sites, there's actually a 16.3% chance you'd see a zinc finger. So they seem to be a little bit overrepresented there. And this is plotted here on this graph. This is just a percentage plot maxing out at 50%. Baseline bimaculoides, 5.3%. Out of our pool of significant edits, the 12,000, there's 16.3%. And then in the 51 high priority edits, 51% of those are zinc fingers. What this means is when you're looking at highly edited genes in response to OA, ocean acidification, in octopus rubescens, there's a good chance you're looking at zinc fingers. So what are zinc fingers? Why would they be upregulating RNA editing in these genes in particular? What do they accomplish? This is a picture here of 3 zinc fingers. A zinc finger is a protein with a short helical region like this, and then a little linker domain that's flexible that folds on a zinc ion here, and then continues on into something else, often another zinc finger. And they're usually found connected to some kind of effector protein because zinc fingers bind DNA, and they can allow another protein to bind specific DNA. Here, this one looks like it binds to a DNA sequence GAG. But if this zinc finger is found with other zinc fingers connected to it, which they very often are, then it might bind some other sequence. So zinc fingers are connected to effector proteins to deliver them to where they need to be they transcription factors or nucleases or repressors. And any one zinc finger won't exactly tell you what the whole protein complex is going to do. Excuse me. So this is a bit of a hard spot for us because we have these edit sites in these open reading frames showing us that these are zinc fingers, but we don't have more data on either side to tell us what are these zinc fingers connected to and what are they in yet. But that's kind of where my experiment stops in that regard. I did try to do other things, though, because I had more than just the goal of see what I find in terms of like editing sites like this. I also wanted to verify these editing sites that we had in these octopuses using a separate method. There was a little confusion in my head originally. I was going to say something about the number of octopuses. So I said we started with, we did, but we only got expensive RNA and DNA deep sequencing for 6 of those. So 3 from the control, 3 from the ocean acidification. And that's what our data up to this point is based on. For any one of the edits that we find, we can then go to the other 6 octopuses that participated in the experiment, amplify out that site, and then sequence it using a different method that is a lot more expensive per nucleotide, but if you're only sequencing a little bit, it's actually way cheaper. And that was what I decided to do for a few different edits. It was originally going to be 10, and then things happened. I chose 10 edits to verify, and this is my process that I used to design primers for them. I would get the open reading frame in a document and take a look at it. I would mark where the edit was. This A was being edited to a G. And then I would use a primer design tool and try and figure out primers to use to amplify this gene so that I could send it off for sequencing. And then it ended up that I had to use 2 sets of primers because I kept on getting interesting results. But I got to work. I did PCR, and then I purified the product, and then I ran it on a gel, and then if it looked okay, I did that again, and rinse and repeat a whole bunch of times. Not the most exciting part of the experiment. And then I had samples ready to send off to Texas for Sanger sequencing. Unfortunately, I did not get what I was looking for. None of the samples that I tried to verify showed sequencing by this method. Now, for one particular gene, just as an example, the pipeline said that this octopus was editing 22% of its transcripts. This one was editing all of them. This one was editing 18.5%. A good bit of editing across the board at this one example edit that I'm showing you. Then when the Sanger results came back for this same edit site, that's nothing. 0, 2, 2, 4, nothing. That's basically noise. What I think probably happened is that there's DNA contamination in the RNA that I isolated, because I tried to get DNA and RNA from each organism separately. If there's DNA in my RNA samples, it could explain what's going on here, not 100%. So I had to move on from that. And as I was trying to finalize my results in the numbers and everything, going back over the editing, was working with Kurt, and we found another interesting trend that we hadn't quite expected, but makes sense. So if we look back, not at our data, but at the temperature-dependent RNA edits in Octopus bimaculoides, and this is a graph I showed before, remember that as temperature goes up, editing goes down? That's what this histogram is showing. So this histogram is showing bars on the right are number of editing sites on the y-axis. So as the change in percent editing, negative and positive is a little bit confusing in this case, but just the main point to take away is that to the right here, there are a lot of edits that are more positively edited in cold water, versus to the left of 0, these are the edits that go up in warm water. And it's very obviously lopsided to the right, meaning they're cold-induced edits by far, not warm-induced edits. I made a similar plot for my data, but this is considering acidity of the water instead of temperature. And this one is also lopsided. It is in the other direction, but it's got the same axes. So number of editing sites on the vertical axis. And sites that are higher to the left are ones that were upregulated in low CO2, or the control treatment, versus to the right, it's sparse. There's lower editing in higher acidity water. So the octopuses that were in the OA treatment experienced RNA editing suppression. They do it less. I should mention, because I've been talking about cephalopods so much, there's a reason for that. When it comes to editing in coding regions of RNA, the main organisms that do this are coleoid cephalopods. So these are the squid, cuttlefish, octopus, but not nautilus, not gastropods. If we look at the number of A to G edits found in the nervous tissue of a few different organisms, there's squid, cuttlefish, another octopus, vulgaris, octopus bimaculoides, we see there are a lot of editing sites in these organisms. That's 130-some thousand there. If we compare that to rubescens nerve tissue, that is still a lot. It is lower, but it is still a lot. It holds with the trend that coleoid cephalopods have a lot of A to G RNA editing, versus the rubescens gill tissue, definitely a lot less. Nautili and sea hares, obviously very low amounts of A to G editing in their nervous tissue. And this is interesting because the coleoid cephalopods diverged from other cephalopods around 240 million years ago. Now, the ocean 240 million years ago was actually warmer and more acidic than it is today. In fact, as our ocean today warms up and becomes more acidic, it's going to more closely resemble the ocean as it did when coleoid cephalopods first diverged from other cephalopods. So this is just speculation at this point, but it is a pretty big deal. It could be that some of the editing that has evolved since that split is an adaptation to the ocean getting colder. I can't say for sure, obviously. It's still early days in terms of RNA editing and cephalopod research, but it's something to think about. Aside from the RNA editing itself, one of the things that was continually a thorn in my side through this is that we don't have an octopus rubescens genome. That would have been very handy to have when doing the pipeline originally. We wouldn't have had to sequence DNA for every single one of the octopuses that we sequenced. We could have just gotten RNA. And also, our method doesn't find anything but edits within coding sequences. So upstream and downstream untranslated regions we're not finding edits in those. We're not finding edits in non-coding RNA. There's a lot that we're missing by not having a genome. Thankfully, later on in my time at Waterloo University in my master's, Kurt managed to get funding for some more sequencing of a separate individual octopus named Aristotle, a ruby octopus, Octopus rubescens. And I took an arm snip from him, and we sent it to UC Davis for expensive sequencing, PacBio hi-fi long-read sequencing, which is very nice to have. And with that, I went through that assembly process that I talked about, like with the encyclopedias and stuff, reassembled the long reads into a preliminary octopus genome assembly. Well, I should mention, I don't need to go into the snail plot. I just like it because it looks cool. I know John likes it. And this is an output that's showing me some statistics about the genome that I assembled. And if you have questions about it afterwards, I'd be happy to answer. I have a little bit more readable results here. It's necessary to consider, while it's a preliminary draft genome, and it's still kind of fragmented, ideally if it were fully assembled back into a reference genome, it would be in the same number of pieces as there are chromosomes in octopus. It's more fragmented than that, but it's not bad. It also doesn't have any mapping or annotations. It's just put together into contiguous segments. However, if you take a look at some comparisons, on NCBI, there are some reference genomes available. In particular, at the top there, Octopus bimaculoides and Octopus sinensis, chromosome-level reference genomes. Great stuff on NCBI. And there's some numbers associated with these, which I don't need to get too deep into, but let's just take a look at the scaffold N50. Briefly, to say what an N50 is, go back to the encyclopedia example again, since it's so useful. Imagine you're reassembling your encyclopedia, and the more you reassemble those pieces that you read in the digital world, the fewer fragments you have because you're taking two and building them into one, contiguous segments over and over again. If you were to is 10 millimeters long, throwing numbers out there, and your fragment. And then you take your next fragment you've reconstructed that is the next longest, say that one's 1 million, and you add that. You have 3 million letters of reassembled stuff, and you keep going until you hit 5 million, which is half of 10 million. The last segment that you've added on to those segments, the length of that is what we call an N50. Basically, the better assembled your genome or encyclopedias, whatever you're assembling, the higher that number will be as a length. So just comparing, the scaffold N50 of bimaculoides and sinensis, 97 million, 106 million, good, high numbers. Mine, 5.7 million, considerably lower, so it's more fragmented. But go back again to the encyclopedia example. Let's consider this number here. Imagine you have, say, 100 entries from your encyclopedia set that you have separately recorded on file. You have perfect copies of them. If you reassemble your encyclopedias correctly, you would expect to find an exact match between those 100 entries you have copies of, and if you do a search through your assembled encyclopedia, those 100 entries in there, if you do it perfectly. If you jumble your reassembly, you might end up only finding 90 or 80 or 50 of the entries that you ought to, and the other ones got corrupted in the reassembly. This number here is something similar, but for genomes. And again, doing a comparison is useful. If they had perfectly reassembled these octopus genomes from their sequencing data, these numbers would be close to 100% for the reference genomes. They're not, but this is a baseline which we can assume is very good. Mine is 87.5. That's not very far off. If you're too aggressive with your scaffolding in your reassembly process, you might end up with something a lot worse while still having a very, very high N50, and that might be suspect, which didn't happen to me, so that is good. Lastly, since my project is over, it's nice to know that it won't end there. Meg Mindlin is going to continue the research. First off, or maybe not first, somewhere, she's going to try to fix my mistakes in the verification process by removing the DNA contamination from my RNA samples, and I really hope that works, because I'd like to see success there. And she's going to take a closer look at the zinc fingers and try and do some computer modeling of them to figure out what they associate with, what their structure is, what their function is. Just as a summary, since we've gone through quite a bit here, I reran JD's experiment, only with nervous tissue instead of gill. I found a lot more editing, which I was glad to, and zinc fingers are highly overrepresented in the edits, which is interesting. I also found editing suppression in response to ocean acidification. I tried an independent method for verification. Didn't go as planned. That's how things go sometimes. And I made a preliminary octopus rubescens genome assembly, which can be very helpful going forwards, not just RNA editing, but also in other applications, which I'm sure Kurt is hoping to get grad students to get working on, like some differential gene expression or who knows. I definitely couldn't have gotten here alone. I, for sure, am building entirely off of JD's bioinformatics pipeline, so big thanks to JD, wherever he is. I'm very, very appreciative to John for always being basically a shoulder to cry on in terms of bioinformatics, because that's about all it was for me. The divers, Emily and Mackie, collected the octopuses at Driftwood Park, and they were rock stars at doing that. I was super nervous, and they were all over it. Stacy was there all summer of 2022 with me and basically did half of the octopus care, even though she didn't need to help me at all. And boy, she suffered for it too. But I'm extremely grateful to Stacy. Nate Iwakoshi, Rachel Doherty were instrumental in helping me not lose my mind when we were harvesting octopus tissue. I'm so glad they were there. And Shane is okay too. So are there any questions?
So do we know if this RNA editing happens in any other species, or is it just in octopuses and related cephalopods?
It's the AG RNA editing in coding regions is selected for in coleoid cephalopods. But there's tons of RNA editing in other organisms. There's millions, or at least like 1.4 million edits in humans, but in non-coding regions. There's evidence of other RNA edits in other organisms that are vital for their survival. Like there's a glutamate receptor edit in mice that if you take it away, the mice die. But it's the only edit that matters in mice. There's essentially selection against edits that change proteins in other organisms except for coleoid cephalopods.
Do we know the mechanisms by which changes in temperature or changes in ECO2 affect ADAR?
I think I read regarding temperature, but I've forgotten exactly how that would work. With regards to ADAR, I could take a stab at like saying, well, there is some evidence in some cases that it could be. There's no direct proof, but there was an organism, well, actually, there's a couple lines of reasoning behind this. There was in human embryonic cells, they looked deeper into the mechanism of ADAR. Basically, to do its thing, there's a glutamate on the enzyme that needs to be protonated in order to carry out the edit. There's a flipping out thing and then flipping in. So in a cell, basically the lower the pH is, the more likely ADARs are to be able to perform their editing function. And there's evidence in, it was a Sepia officinalis, that lowering the pH of its environment can lower the pH of its cells. But those are kind of two separate things. You can draw a line through them if you want as evidence for a mechanism by which change in acidity can affect RNA editing. But we're lacking a lot of data.
I have one online from JD here. He says, you mentioned you assembled a better rubescence genome. Have you thought about rerunning the pipeline with a better genome transcriptome?
Well, if I had the chops to do that, I could fairly trivially like swap out the alignment command in Bowtie from using the RNA transcriptome that I made and instead using the assembly, but I'm just not exactly certain how it would play out or what it would look like or what I would then do after that. But if JD says it might work, maybe it might work just that easily. Maybe I should do that. Any other online questions?
That's a good one so far.
Silent.
For the continuation, will it require a genome or
There's tissue frozen. There's still some left, right? Yeah, there's
We may have to get like one extra octopus possibly.
I'm not sure how that's going to play out with replicating it very exactly, but
I think we just need more sample. Like it's not necessarily replicating the conditions. It would be specifically for characterizing the zinc fingers.
oh well, that yeah.
So we use the tissue to cell culture.
Oh, that'd be cool and stuff. Yeah, but like for the purposes of this, it's not all lost anyway. Yeah. So hopefully not. Should I have repeated that to the?
Yeah, sure.
He was asking if we need to collect more samples from more octopuses in order to continue, and then I answered, we have stuff on ice, hopefully, from that experiment. We took extra. Any other questions?
Not a question, really, just a comment. You mentioned protein motors early on in the lecture.
Kinnison?
Kinnison, I think actin
Kinnis,
associated with those two as far
yeah,
as the strands that they travel on.
the actin microfilaments, microtubules. I need to,
I believe
yeah.
that there, Ron Vale, I think his name is, has some really nice lectures online regarding those, and they're really quite well done and quite interesting.
I should probably go back and review those.
You're talking about the bioinformatics process where it would have benefited from having a full genome for octopus rubescence. I mean, was that pretty expected that there wouldn't be a full genome available? I mean, I don't know how common that is for full genome sequences to be available for any wildlife. Is that pretty expected that not be available? Or?
His question was, did we know beforehand going into it that there wasn't a genome available, and how did we navigate that? We definitely knew, and we've all known that there isn't a rubescence assembly because there hasn't been the money and willpower to make that, and that's a common and difficult thing to deal with when you're not researching yeast or worms, but it is the way it is. There aren't great assemblies for most animals. It takes a lot to get good assemblies. But we're a lot closer to having one for rubescens than we were.
Okay, going once, going twice. Okay, thank you very much
Thank you. I know, it's probably really boring.
I like that.
Good.
If you have enough intelligence to stay along with you, it's pretty interesting.